Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. And you are watching Bioinformatics Insights. Today I'm going to start a new series, a kind of course, and its name is Python for Biological Data Analysis. So let's talk briefly about this course or this series. I have two goals for this series. The first one is to equip biologists with essential Python programming skills for data analysis and scientific research. That means if you are working in biology, whatever micro, biochemistry, biotechnology, or any other related field, botany, zoology, so you are welcome and this course is for you. But if you are a student of computer science, so I'm afraid this course is not for you. The reason is that I will use all the examples, each and everything from the background of biology. I will not use the background of computer science and any other thing, so it may be boring for you. Okay. And goal two is I will emphasize on practical exercise and real world biological data sets. How I will emphasize on the practical data sets or the practical example because I will teach you in Colab and you can also practice using my Colab or your own Colab for learning. Okay. What will be the contents of this course? So apparently I will cover 10 videos for this course, could be 11 or 12, but 10 videos are essential. The first two lectures will be foundation and about control structure. The next two lectures will be about data handling, data structure for biology and working with different files. The next two lectures will be about scientific computing. I will discuss the use of NumPy, pandas, and data visualization. And then the next few lectures will be for sequence analysis, statistical analysis for biology, and some practical projects. And the last two lectures are optional, but I will cover that will be advanced topics in like bioinformatics libraries, how to use this Python for bioinformatics. So that was all about this course, this introduction. So today we are going to start our chapter first, and its name is Foundation of Python. So let's start. How will you start it? Let me show you if you are new to my channel. So you have to write or you have to type colab.research or let me do like this. Just write colab or simply just write Google Colab. Let me teach you this way. You will have this thing in your computer. Okay, click here. Once you click here, now you need to click GitHub. So once you click the GitHub, type here ASH100, H100, and now click this option and scroll down and select Python for life sciences or life science. So far, I have managed two chapters. So today I will cover the first chapter, the foundation chapter. So you need to click here. Okay. You will have this kind of window in front of you. So today we will cover all this, a kind of founding or foundation chapter. Okay, now you can click here and the notebook will be connected. This is all about some introduction of myself and about this course, a brief one sentence introduction. This is the course outline. You can watch it chapter wise, you can read them. So, today we will cover the foundation, and in foundation, we will cover basically these things. Okay, so scroll down. Okay, so let's start. Getting started with Python and Colab. What actually is Colab? One kind of disclaimer. I will not go for an extra detail because I will try myself to complete each and every video within 30 minutes. So that's why uh, I will try my best to wind up these videos, each of my video before 30 minutes. Okay. So why we will use Colab? This is the Colab you can see. So there are many reasons because it's very easy for everyone to navigate to Colab. It's also very easy to write a basic script and to practice your scripts, or to practice my script and repeat my script or generate your own script quickly in the Colab. And you can also work with different data types in Colab, so no boundary issues and no other legal or illegal issues. Okay. And understand variables and basic operations. I think it is very easy to teach or to practice the new user in Colab, because most of the people or the student do not have a sophisticated systems or sophisticated operating systems. So that's why Colab is the best. Okay. So this was all about the introduction to Colab and why Python for biology. This is part two. Actually, using Python, we can analyze biological data like DNA, like RNA, or I would say biomolecules like proteins and many more. We can also process experimental data. If you have some experimental data from your lab, so you can also use Python and to perform different analysis. You can create publication quality graphs very easy on your fingertips, and you can also automate calculations. For example, you have some formula of molarity or solution preparation, or from population genetics or something else formula if you have, so we can quickly write here and we can perform complex calculation within Colab, and we can save your Colab and you can share it with others like I'm doing. I'm sharing or I'm teaching from my Colab notebook. Okay, so let's start your first Python code. So, what will be your first Python code? Let me also tell you, you will see this play sign in some areas like here, like here. So, actually, when you click here, so you will actually, you will in reality, you will run the command mentioned here. In case you want to generate this command line here, so just keep your cursor here, you will see code and text. Let's say I'm clicking code, so I created an empty space and I can write again. Okay, so let's say there is there is a function in Python, so let's start from the print function. There are many words like functions and other things. We will learn it sidewise during this course. So don't jump to each and every word I use from now. Okay, so print is a function, and if if I call it a function, so we will anything is a function. We write like this: the function name and the small parenthesis. So in this case, it's a print function. So you are asking Python to print something. So what to print? So here you can see print welcome to Python for biology. But if you see carefully, welcome to Python for biology is enclosed in double inverted commas. For example, let me write again, uh, welcome to Python for biology. So the difference is that here you can see the inverted commas. I play here. Okay, okay. I play here and let me play it again. Okay, clear. I play and you can see welcome to Python for biology. But in this case, it provides an error that invalid syntax. Perhaps you forgot a comma. Of course, I forgot an inverted commas, just like I put it here. So the first thing you learn, like to using the print function and enclose your statement in the inverted commas and you can print it down. Okay. Now, section 2. Okay, there are so many other functions too, but I just started from the print function. Okay, section 2. Okay, no, sorry, section 3. Comments documenting your code. Comments are text that Python ignores. They are for humans to read in research. Good comments are essential. For example, that means that, for example, if you write some code and you want to write something for that comment, let's say I'm writing a code like close. For example, I'm writing print and I want to write like let's say DNA is made of nucleotide. But let's say I'm not a biologist and I will present this piece of information tomorrow somewhere, but I don't want to forget this name nucleotide. So I will write a comment for myself like nucleotide. Let's say I want to know about the base pair. So for example, I tomorrow I want to mention about the base pair. So therefore I can drop this word base pair in the comment in front of this code. So now if we run like this, it will provide an error, invalid syntax. But if we pro we use this comment in a proper way, so how we can use in a proper way? When we use a hashtag sign, and now you can see the color is green. So now you will see DNA is made of nucleotide, it ran successfully, and this is ignored by the Python because it was a comment. So the same way you can document, you can drop some comments with your code and using the hashtag sign. When you use the hashtag sign, it will not run. The program will be unable to run it. Also, the program will not provide or throw any error in front of you. Okay, this is a okay. This is a single line comment. Okay, now we are moving towards some next thing. Uh, what are variables and how we are using the variables? But okay, that will be we will discuss it here. Let's say learn another thing. For example, you can see organism is equal to E. coli. Okay, the same way I use print, but here I mention organisms is equal to E. coli, temperature is equal to 37. Print growing organism at temperature degree C. Just pardon. Okay. Now, I first asked the program to memorize organism is equal to equali. And remember, if you can see, I enclose this word in a uh, double inverted commas. And then for temperature, I did not enclose anything. I just put 37 because we will discuss later. This is an integer and this is another different data type. So for different data types, we will use different methods, and that are simple. Okay. So organism E. coli temperature 37. Now, if we write print and before we mention like the print command like this code will run. This was our print command or the explanation to be printed. In this case, I'm using print, and once again, I use the same parenthesis, but here I mention the F, and then I inserted the growing word inside the commas, and then organism at temperature degree C. So let's play it. Oh, it says error. So why it's error? Wait, I think one comma was inverted comma was missing. Okay. Print if growing normal at temperature. Okay. Organism. Invalid character. Okay. Got it. Okay. Okay. So because when I'm teaching, I forget many things. I was running this command, the print command, but the computer or the program did not know what is organism and what is temperature. That's why it throws an organism error. So first we will run this. Organism is E. coli, temperature is 37. Now if you see print, and this is we call it the formatted command or whatever the if is for format for, if we are interested or if we are mixing different words with each other, so we will use this, but you will learn this later. So print f, then growing organism, but organism is also defined from the organism, it means E. coli, and from the temperature that means 37. So when we run it, it will print growing E. coli at 37 degrees C. Let's say I run another thing, boy is equal to David, age is 25, country is USA. So first run this. Now we will use the print command, print it, and it's a function, so the parenthesis will start, and then this inverted commas will start, and boy is of age year old living in country. But all these three word boy, age, and country, they will be enclosed in a curly brackets. So let's click here. So it says David is of 25-year old living in USA. So actually this command, country, age, and boy, they just translate it into their values. The boy value was David, the age value was 25, and the country value was USA. Okay, now we are moving to variables and storing your data. Like we are using variables for storing our data. So what actually are variables? So Variables are just like the labels. For example, you're working in the lab and you have 10 test tubes and you are performing your experiment. So in one you just mention like DP1. In another one you will mention like concentration of NaCl this much. In the third you mention 0.15 micromolar or millimolar, whatever. So those mentioning are actually the variables of u. So after 3 hours, if you need a solution of 0.15 micromolar in HCl, so you will just see, okay, this one. You will pick this test tube. So because that labeling was actually the did the trick for you, and that you can say that variables are the same thing as label in the when we are using in our labs. So but there must be some rules for the variable or for those labels. I will call it labels. They must start with a letter or underscore. You cannot start a variable with a digit, let's say 1, 2, 3. It will throw an error. It will contains letter, number, and underscore, but the numbers can be contained in the middle, but not at the end, at the start. Okay, these are case sensitive. In variables, the DNA is not equal to DNA. Yeah. That means, for example, if I say that DNA is equal to, let's say, wait, DNA is equal to, let's say, um, E. coli, let's say I'm running it, and now if I say that DNA is equal to, let's say, is RES. So these two are different variable because of the this is the uppercase, this is the lowercase. So that's why I mentioned like case sensitive. And you cannot use a space in like let's say DNA underscore 1 is equal to E. coli. That works fine. But if you use a space, DNA space 1, you can see invalid syntax. So point I want to mention is that while putting a variable or while putting a label, I will use the name. You cannot use space. And once again, I most of the things I already explained, that variable is an m that store a value in a memory, so you can use it later. Think of it like a label container that holds data. So, for example, gene name BRCA1, patient age 45, DNA concentration 2 is mutation present is equal to true. So these were all the variables. You can use them, but it has some rules I mentioned above, like do not start it from the the numbers. Okay. Uh, what are the example of poor variables? So try not to use, for example, X is BRCA1 2 gene. That will be wrong because it will throw an error. It start with 2. And gene name, because I said gene dash name, I already mentioned you can use underscore but not dash, so it will also generates or throws an error. Okay, so the, you can use, then later on we can use the print command. Let's say print gene and gene name. So if you see here, the gene and the colon is enclosed in a double inverted commas, print parenthesis, double inverted comma that contain gene colon and then comma gene name. So the gene name is already explained here. That is BRCA1, BRCA1. So if we print only this. Let's say if I, yeah, it's already here. Print gene name. It says gene colon and BRCA1. So that is the use of that, how you will create different variables and how you will use the print command in simple operations. So for the other things, you can do it yourself because the time is running short. 25 minutes, and today I will go possibly for 35 minutes because in the start I talked about some introduction of this course, so that's why. And also this is my first lecture in this series, so I want to catch most of you people to transfer you the interest and the concept. So that's why I will take almost a 35 minutes. Okay. Not you can perform some variables, create variables for your own biological data. For example, species name, sample size, and experimental date, and then you can use these lines of code. If you need it more, just generate it. Click. You can generate more. So you can create some of the variables right now while doing this exercise, and then use the print command the way I use it here, and let's see is it working or not. Okay, now we will talk about some data types. I mentioned in the start that there are different data types, so we will talk about some of them. So, what are the data types? For example, for us, it is very simple, like numbers. When we speak, we differentiate that what is your mobile number. So, we put the word number. It means we are asking for a number for a specific type of data. If I ask, what is your mobile? So maybe I can receive an answer. My mobile is Samsung, or my mobile is Android. So many different answers. So actually, if while we are speaking in our daily life, we are also using the data types. We are classifying things. What is the name of your mobile company, whatever you say? So then the person will not tell us their number. They will tell us the company name like Samsung, iPhone, whatever. Um, the same way, when we are dealing in Python, so we must have some different data, or we have to classify, we have to understand these different data types. So we have integer, so the integer will be a simple 5, you can see, and it will be a whole number. I think you should know a whole number. What is float? If the data contains, if the integer contain a decimal, so it will be a float. It will be called a float. What will be string? String will be a kind of text or sequence. It could be a DNA sequence, it could be a sentence, it could be a RNA sequence, so that will we will call it string. What will be bool? Okay, or boolean. So the bool will normal when we use true or false, so we call it logical values or Boolean operators. So in representation we write it bool. So bool will be always true and false. What will be the list? For example, you have some data, it has many names. So all those names separated by, let's say you are entering the your class fellows, your variable name is class fellow. So you are writing John, comma, Ellen, comma, Alex, comma, David, comma. So these all are this we will call it a list, and that will be just some mutatable collections of in some specific order. What will be the tuple? And that will be the ordered immutable collection, but we will talk about this in chapter 4 and 5 in detail, so I'm not going in much detail here. What will be the dictionary? So that could be a gene like gene BRCA1 and key. These are also a key values. The dictionary will be a pairs of values. Let's say gene and BRCA1. We will also talk in detail later. And a set, and the set will be an unordered or unique values number, so we will also talk later on. But for the initial stages you should know about the integer, float, string, boolean and list. Okay, so now let's use this thing in uh in biology. Okay, so for example, we are using a string. So what was a string? I told you a string is a kind of sequence or a data. So first I generate a variable species is equal to Homo sapien. DNA underscore sequence is my second variable, and I drop some sequence here ATC, and you can read the sequence. Then experimental note, I added an experimental note like sample collected from healthy tissue. Let's say I run this cell which contains, so I generated these three variables, and now if I print the species, it will say, if I print the, okay, if I experimental note, okay, so you can, I here I use just the print for species, so that printed the species name. The same way you can print, let's say I'm using the print for this one, print, and DNA sequence. Okay, nothing else. Let's say, oh wait, this is print, let's say uh, my sequence, my sec. This will be, I want this name and then comma, and then mention DNA underscore sequence and close it. My sequence colon, and for the DNA sequence that print the value of the DNA sequence which we stored here. Okay. Now we are coming to integer. So as I told you, integers are the whole numbers. So let's say chromosome, a variable 46, patient count 120, generation number, whatever, let's say 5. And you can first click here that the computer or the Python should store it. Now you can print these information as it is. Now we are coming to the float, and float will be that integers which contains the decimal. So once again, you can run this and you can print them. You can see now I just manage the, like you can also do like this. It is not necessary like to write the print for each single variable. You can use the same cell and print all your variable like I did. So because I will give you the advancement just like slowly and steady. Okay, now coming to the Boolean, as I told you, the Boolean informations are will be in true or false. So it's healthy and true and false. So the information are stored here. Now you can print again. So you can also try the other things. Now we are coming to some basic operations, and I will be quick here because you now understand what are variables, what are print functions. So in arithmetic operations, we will memorize a kind of different formula to inside the cell. Let's say my first variable is sample one cells, this much, sample two cells, this much, and then total cells is equal to sample one cells plus sample two cells. When you run it, you will have now it ran successfully, but if you print the answers. So you will have the answers. These are the sample one cell, these are sample two. But what operations I perform, I calculated the total cells very easily and very quickly. More arithmetic operations for bacterial arithmetic, for bacterial growth calculation. You can run these cells because I'm going a bit faster now. Okay. Now let's say what kind of string operation we can do. Strings, as I told you, strings are kind of sequences or letters or words or sentences. So the gene ID I mentioned here, the gene sample I mentioned here. Now I need kind of full identifier is equal to gene ID plus, now insert this underscore plus gene symbol, and you will see full gene identifier in this thing. Okay. String repetitions, you can do it. You can do it yourself. It's easy. Now, print statement and displaying your result. As I told you in the start, that the print is actually a functions that your main tool that actually displays your data. Okay. So remember, in Python, there is not a single function. There are so many functions we will use during this course, and you will learn so many functions. Even you can use the Google to take an appropriate function, and you can directly use them. Python use its own functions. Okay, so it will not be difficult for you. So I would suggest you run it. Okay. And then finally, I drop some intricate exercise, like create your lab profile, create variables of your information, and print them. You can increase these variables, and then you can um, edit this data. You can add more variables and then print it as usual. And also you can uh, perform some for exercise. For example, DNA underscore sequence, we ask the program to memorize this sequence, or we generated this variable DNA sequence, and then we say calculate and print the following: what sequence length. Okay, once again you can see len and then DNA sequence. So actually you can see the information here. Len is a built-in, another built-in function that calculates the length of an object or the item. So in this case, what will be the length of our string? So you can see length is 21 base pair so quickly. Okay, now in second operation, you can count the ATCG and then print it like 5A, 5T, 6Gs, and cytosine 5. Also, here we calculate the GC content. So, because Python do not know the formula, so you will just write the formula here, and then it will be able to calculate your GC content for your that sequence. So this was exercise 2, and this exercise is from the lab measurement, so I will leave it for you. And finally, there are some common mistakes, so try to avoid it. While creating the variables, try do not forget quotes. If you forget quotes, it will give an error. You can say name BRCA1 is not defined, because if I mention a quote, let's say, not quote, the like the inverted commas. So now it is fine. Okay. Another thing, there are some built-in functions. For example, if you are writing some built-in not functions categories, mammalia. So mammalia is a built-in. It will generate an error in valid syntax, because the mammalia is uh, stored by the organism class. So if you write If you see, let me turn off this as I mentioned as comment. As I told you in the start, if you put this sign, it will turn off the code, and I'm turning on this code, organism underscore class. So now it did not generate any error because Mammalia was actually saved already with the organism class. So there are certain things which are already saved with Python. So Mammalia is one of them. Okay. So once again, as I told you that it is case sensitive for a gene with capital G BRCA1, this will be a different variable from this gene with the lowercase and lower BRCA1, this will be a different variable. Okay. And mixing data type incorrectly, this will also provide an error. For example, if you're mixing boolean with integer with some strings incorrectly, so it will generate errors. So you will understand these things with the passage of time, because we will cover 12 to 14 lectures, I guess. So you will understand these basic things with me. And this is some practice problems, and I leave it for you, and it will be also present in the at the end of every chapter, so you can perform it in your home. Okay. So summary and next steps. So congratulations, you have completed chapter one, and you learn how to use Google Colab. You learn how to perform some basic syntax operations. You learn what are the different types of variable, what are variables and what are different data types. You learn how to perform some basic operations, how to use the print statement, and how to avoid some common mistake. So that's enough for today. And for the first lecture, please give me a thumbs up if you are liking my lectures. And I'm sorry, it may be a bit boring for you, but if you have an interest, we will carry on, and you can learn with me something, if not all, but at least I can provide you a foundation stone in Python. So, see you then. Bye bye.